great pleasure, I will introduce today's speaker, Dr. Fred van Leeuwen. Fred did his PhD with Piet Horst. He studied base J in trypanosomes. And after that, he moved to Seattle, where he joined the lab of Dan Rotchling at the Hutch for a postdoc. And there they just found genes that were regulators of telomeric silencing, but little else was known about them. And Fred found that disruptor of telomeric silencing 1, or better known as Dot1, was a very special methyltransferase, methylating histone H3 and lysine 79. After his postdoc, Fred moved back to the Netherlands to start his lab at the Netherlands Cancer Institute. The love for Dot1 remains, and he's really greatly improved our understanding of it. But he's also made very important contributions to the field in other aspects. For example, histone inheritance, and more recently looking at chromatin composition. And he's often used very clever, very creative methods that let him address questions from a new angle. So it's well deserved that last year he was promoted to full professor at the University of Amsterdam. So I should wrap up, but I just want to end on a personal note because I was lucky enough to do my PhD in Fred's lab and learn a lot from him. And he's been a fantastic mentor to me throughout my PhD and to this day. So I'm very happy that he'll give the seminar today. So with that, Fred, go ahead.
Okay, thank you, Hanneke. Yeah, thank you all for joining this seminar and welcome to my talk. Thank you for making me part of this great new community in these strange times. I discovered a Discord server with lots of exciting science going on. So yeah, happy to be here. What I would like to start with is an ambition that was expressed recently by our Chemistry of Life Society in the Netherlands, where they said, what we want to do is build a molecular encyclopedia of life. And the idea is if you map all the molecules of the cell, then you can influence their function, interactions in a rational way. And to rephrase this, that if you really have a good fundamental understanding of a molecular level of life, then you can intervene and adjust in a targeted manner, and you can contribute to the grand societal challenges that we have at the moment. And I really like this because it says that if you do good fundamental science, you will make important contributions, and I think this is true. And for this crowd, I think we can make this a bit more focused and say, and as this is something that we as a lab want to contribute to, is maybe make a molecular encyclopedia of chromatin, right? And that if we map all the chromatin molecules and know how they work, then maybe we can use that to change the behavior of a cell, or maybe design a cell in a smart way. And this is maybe a bit less ambitious, but we can still contribute to solving these big problems. I'm convinced of that. So this is a goal of my lab to see if we can make a contribution here, and maybe to make it a bit more concrete. Then what do we need to achieve that? So we need to be able to measure, perturb, and predict, and these are really exciting times. Like measuring chromatin is now possible in many different ways. There's new technologies popping up at a rapid pace. And this is, I mean, it's fantastic to be part of this at the moment, right? And I think this will allow us to measure chromatin and their proteins, different conditions, different times, but also different scales from a structure to, something that happens in a cell or an organism. Also perturbing, we can do a lot at the moment with protein and genome engineering methods, drugs that are being small molecules that target these chromatin proteins. And I think if we know, if we develop measuring and perturbing to a high level, then in the end, we will be able to predict exactly, what happens if we poke at a cell, and we know what the output will be. And so my talk today will be, in essence, about different ways to measure chromatin, about 2 technologies that we developed in my group to measure chromatin structure in a cell. And the middle part of my talk will be about trying to perturb one of these chromatin factors in mouse using kind of simple technology, trying to address the function of this chromatin factor. And so I think the field has made, enormous progress in the last years, decades. And we're really good, I think, in annotating the core components of chromatin. And this is an example that you see often in textbooks. And these core chromatin features You know, they are well known to most of us, and they're super useful for annotating the genome. You know, it's with these marks we can find enhancers, promoters, we can define areas of heterochromatin. So this is a great starting point for our encyclopedia. But it is more because we also know what these marks do to some extent, and we know how to manipulate them because we know enzymes that write these marks. There's demodifying enzymes. There are protein domains that read these modifications. We know about how they collaborate with transcription factors. There's small molecules that can interfere with all three of these protein families. So I think we're in a really good place to make progress in understanding how this all works together. But of course, there is much more than these core components. And it is also clear that of these other components, we know much less than all these factors. And this is the focus of today's talk, is this one protein histomodification. It's methylation of lysine 79, which is located on the core of the nucleosome. And it's introduced there by Dot1 in yeast or Dot1L in mammals. And this methylation system, like Hanneke mentioned, was discovered almost 20 years ago in budding yeast, and then rapidly after that in humans. And it took a bit of time before Dot1 was discovered because it doesn't have a set domain, and the set domain is the catalytic domain of all these other histone lysine methyltransferases. But the fact that Dot1 doesn't have a set domain also made it an appealing target for Dot1 for inhibitors, because an inhibitor of Dot1L actually is very specific for Dot1L, and it doesn't target these set domain proteins. So Dot1 has more like a class 1 methyltransferase domain that is found, for example, in arginine methyltransferases or DNA methyltransferases. And Dot1L actually became famous when it was shown by several labs already quite early on after the discovery that in a certain type of leukemia, where the MLL protein is rearranged by genomic translocation, that these leukemias, the MLL rearranged leukemias, they are dependent on Dot1L activity. That means that they are sensitive to Dot1L inhibitors. And that is not because Dot1L is mutated, but because Dot1L is recruited to the wrong place in the genome by the MLL oncogenic fusion proteins. And so what do we know about more molecular aspects of Dot1 and Dot1L K79 methylation? Well, it methylates gene bodies, and I'll show you examples later on in my talk about yeast and in mouse. It's a co-transcriptional modification. It happens in gene bodies because it's recruited probably by POL2, by histone acetylation, and also by histone ubiquitination, especially H2B monoubiquitination at the C terminus of H2B. And there's a whole body of work that has studied this, but I refer to the talk of Cynthia Wahlberger at your in this group here, and she has done a beautiful job in explaining the crosstalk mechanism and also explaining it at a very molecular level. So like I said, Dot1L in mammals is recruited by histone acetylation, and I think this together really explains why it is predominantly found in gene bodies. But that's really it. You know, we don't really know then what happens. You know, Dot1 methylates gene bodies. What does it do? What is the effect of methylation on gene expression on chromatin? And we know so little because there are no clear reader domains that translate this into function. And also, so far, I think there is little evidence that erasers or demethylases play a role in this system. And so this is then where we are. It was discovered a long time ago. It has clinical relevance, but yet we know very little. And as my student Tom recently coined, he said, yes, Fred, Dot1 was discovered 20 years ago, but what do we really know about how this works? Despite the work of Hanneke in your lab, you know, we still know very little. And so this really encouraged us, this problem, to think about new ways to look into Dot1's function. And so my talk will be is composed of 3 parts. I'll briefly talk about a method, Epi-ID. It's a new method to measure methylation in thousands of mutants in parallel. Then I'll talk about what we discovered about the role of Dot1L in mouse T cells. And in the end, if we have time, I'll briefly give you an overview of a new method that we developed to decode local chromatin proteomes in budding yeast. But let's start with the method we call Epi-ID as a method to systematically measure methylation in thousands of mutants in parallel. And this project was carried out by Hanneke Vlaming when she was in my lab together with a great technician, Tibor van Welsen. Okay, so before I explain you the screen and show you the data, I want to bring you up to speed in what we know about K79 methylation in budding yeast. And so we measure the methylation in a bunch of mutants with more or less Dot1 by mass spectrometry, and using that as an input for a model, we built a simple model for how the methylation changes when Dot1L activity or Dot1 activity is higher or lower in a yeast cell. And so this model is as follows. There is a synthesis of histones and deposition, and then Dot1 methylates unmethylated lysine 79 to mono, di, and tri. And if you measure in a wild type yeast cell, the most abundant form is the blue one, trimethylation. There's dimethylation and mono, and a small amount of unmethylated K79. And so now if we increase Dot1 activity, then we see more trimethyl, less mono. And if the activity is decreased, the trimethyl drops and the monomethyl increases. And if there's no Dot1 at all, then all methyl forms are gone. And so we kind of use this, this idea to measure methylation. And also if you do a chip sequencing and then look at all the genes and rank them by their trimethyl over monomethyl score, what we see in the genome is that there are, most of the genes have trimethylation in their gene body. And there's some that don't, and the ones that do not, they have monomethylation in the gene body. So these marks are kind of mutually exclusive. And so in our screenings, we have used this trimethyl over monomethyl as kind of a methylation score to kind of rank all the different mutants. Okay, so now if you look in the yeast genome chip sequencing and you look at how does the methylation of K79 correlate to transcription. And here the genes are ranked by transcription from low to high. And then in essence, we see three kind of classes. So most of the genes are in the middle, where they have high trimethyl, low monomethyl in green. And just focus on the solid line, and this is the wild type cell. There's a small class of genes that is really highly transcribed, where the methylation is actually lower, less tri, more mono. And also at the left end, there is a lowly expressed class that is lower trimethyl and high mono. So there is no clear correlation between transcription and methylation. And we wanted to understand why this is. And so this is why we developed a new method to measure methylation in all yeast knockouts. And we couldn't use a simple reporter assay because Dot1 in yeast doesn't really affect gene expression in a prominent way. So we needed a mechanism, a measuring system to directly measure methylation in a yeast cell at the chromatin level. And for this, we took advantage of yeast genetics and also yeast resources that were available in Canadian labs of Corey Nislow, Charlie Boone, and Brenda Andrews. And these are two resources. This is a collection of yeast gene deletion mutants, knockouts, about 5,000. And there was a collection of yeast barcoders that are all wild type yeasts, but they have at one common locus, the HO locus in chromosome 4, a pair of barcodes integrated in the genome, flanking a selectable marker. And so we used these two library collections, crossed them together using robotics and yeast genetic tricks that I will not explain in detail today. And in essence, what we then ended up with is a collection where we have in every colony, there is a one gene knocked out, and it is also contains a unique pair of barcodes. So in essence, if you would take a colony and sequence the barcode, you then know which knockout it is, right? So they're on different places in the genome, but they're in the same cell. And so if now say we want to look at K79 methylation, we have, for example, a Dot1 knockout there where K79 methylation is absent. And we have a SET1 knockout. This is lysine 4 methyltransferase, MLL homolog, where K79 is still present. And so what is important here is that these barcodes are in the genome, but they're actually flanking a POL2 gene. You know, it's the HO locus and the selectable marker. They basically are random, average POL2 loci. So the barcodes are in chromatin. They're also methylated by K79. And so now we would like to interrogate all these mutants in parallel for their level of K79 methylation. And this is possible because we have barcoded yeasts. We can now pull them together in a flask. We then cross-link the chromatin. We break the cells, and then we perform a chip for example, K79 trimethyl. And in this chip, we will immunoprecipitate chromatin that contains barcodes and which the chromatin is methylated. But the chromatin that is not methylated, that barcode will not be immunoprecipitated. So then we will purify the DNA, amplify the barcodes, and then sequence the barcodes and count them. And then in the end, we end up with a barcode count for every colony. And the barcodes counts, in essence, then are informative about the level of methylation. So for example, in a SET1 knockout, we have a high barcode count. And that means that barcode was precipitated. But in a Dot1 knockout, the barcode count is low, and that means that barcode was not precipitated. And it didn't have K79 methylation. And for example, if we have more Dot1 activity, we would have more trimethyl and the barcode count would be higher. Okay, so this is an example of 2 knockouts. We want to look at all knockouts, and this we did. We measured trimethyl, monomethyl. We also measured H3 chip, and we also took an input DNA of every yeast colony as we expanded them in the culture. And this barcoding of genomic DNA as the cells were expanded enabled us to do a growth curve for each mutant, because the relative barcode abundance is a reflection of how fast a cell grows in the population. And so when we first looked at correlation between growth rate and K79 methylation, and here we used the trimethyl over monomethyl score, growth rate is on the x-axis, we noticed that there is an inverse correlation between growth rate and methylation score. And this is also highlighted on the right here. If we take the slow grower, the slowest growing cells, the 10% slowest growing, that these on average have a higher methylation than the regularly growing cells. So clearly there is a bias in this experiment. And for the rest of the screen, what we did, we corrected all the methylation score for their growth rate, because this is a confounder that is probably not caused by specific chromatin effects. And I just want to explain that this makes a lot of sense. Because in yeast, and actually in most other organisms, there is no evidence for a demethylase. And that means that histone methylation is a very stable mark, but it will be lost by dilution, right? So we have histone synthesis, that new histones that are methylated. The only way to get rid of them is by dilution of histones by replication. And so if there is fast replication, dilution will be faster. If there is slow replication, this dilution factor will be slower. There is also dilution by histone turnover. And in fact, we found in our screen, but I won't show you this, factors that if we block histone turnover, replication independent turnover, then the methylation increases. So it is now clear that histone inheritance and replication independent histone turnover, they have an effect on the H3K79 methyl epigenome, if you will. And we found this in yeast, and this was really gratifying to see that the Crabtree lab recently published a paper in Molecular Cell where they show that this also applies to K79 methylation in mammalian cells. And so if we now go back to this transcription versus methylation question, I told you that at the highest, the most highly transcribed genes, they have lower trimethyl and more mono. It is now known that highly expressed genes have more histone turnover, replication independent turnover. And it is actually quite likely that this explains the loss of methylation. There is simply a loss of methylated histones, and then the methylation cycle has to start over. And this leads to more mono and less tri. Okay, but let's focus on the other parts of the screen. And so we, like I said, we measure histone methylation at these barcodes, at a promoter barcode, and there's a barcode here at the terminator of this locus on chromosome 4. But they follow this general rule of, methylation dependence on Dot1L activity. And here I give you an example of the raw data of such a screen, and these are the barcode counts, log2 barcode counts for monomethyl over histone H3 on the x-axis and the trimethyl over H3 on the y-axis. And so most mutants are in the middle. This is this black cloud. They have an average K79 methylation profile. A Dot1 knockout that is barcoded has no mono, no tri, and a Dot1 overexpressor has high trimethyl and less mono. So this makes sense. And then there is this cloud that is really obvious. Here, there is more mono and less tri. So this is a low methylation score group. And this group they are all known factors. And this is this ubiquitin pathway that I talked about. It is the PEF complex. Karen Arndt, for example, has studied this extensively. That leads to recruitment of the H2B ubiquitination machinery. There's a net A complex that also influences this. And if you lose this, methylation levels drop. If you lose the deubiquitinating component, the methylation levels increase. So with this, one tube experiment, in essence, we were able to recapitulate this mechanism of crosstalk that was known in the field. We also found unknown factors, of course, and this is the most exciting part. And so one unknown factor that I think was very cool is a metabolic enzyme. It's called adenosine kinase. And originally it didn't make much sense to us, but soon after we realized that adenosine kinase is involved in the recycling of SAM after the methylation reaction. So when Dot1 uses SAM to methylate, it produces SA as adenosyl homocysteine. And this SA, if adenosine doesn't phosphorylate, if adenosine kinase doesn't phosphorylate adenosine to AMP, it leads to an accumulation of SA. And SA actually is an inhibitor of methyltransferases, including Dot1L. So this is really exciting that we can find both trans factors and also direct chromatin factors. In addition to adenosine kinase, we also found several components of the RPD3L complex. RPD3 comes in two flavors, a small and a large complex, and it's the large complex that is known to bind to promoters and repress their transcription. So we looked into this a little bit further. By mass spectrometry, we could confirm here that mutants of RPD3 and SYN3, they have more trimethyl. The blue bar is larger compared to wild type. And so this really confirmed our Epi-ID barcode count data. And when we look at chip sequencing, we could actually find out where in the genome RPD3 affects K79 methylation levels the most. And so here you see data of wild type cells, where on the right we have MRP51. This is like a regular yeast gene, where there is high level trimethyl, there's H2B ubiquitin, and there is not so much monomethyl. And there's this odd gene, this is a small part of the genome where there is low trimethyl, more mono, and also low ubiquitin. And what we discovered is that actually these genes that have low trimethyl K79, they are RPD3L targets. Because if we delete RPD3, what we find is that the low trimethyl becomes high trimethyl, and the high mono becomes low mono, and ubiquitin that is low becomes high ubiquitin. In essence, a gene that was odd compared to the rest of the genome now looks like your average gene in the yeast genome. And so RPD3 is a negative regulator of K79 methylation. And what we have shown, and you can read more about this in our publication, that this really involves the activity of RPD3, the deacetylase activity. It probably involves recruiting RPD3 by transcriptional regulators. And we believe that this is a transcriptional repression mechanism that keeps H2B ubiquitin low, also at these target genes. And so now we are in this transcription methylation correlation on the lower spectrum of transcription, where we found that most genes are here, but there's a small number of genes that seems to be more dependent, say, on transcription than others. And our study now shows that RPD3L complex really plays a role in keeping methylation low at these lowly transcribed genes. Another group is the SIR complex that also keeps methylation low at their target genes. Okay, and so then to wrap up this part, we found that RPD3 seems to negatively regulate K79 methylation in budding yeast. But these proteins are both highly conserved in evolution. Dot1L in mammalian cells, we talked about this, and homologue of RPD3, one of them is HDAC1. And so we wondered, could this mechanism of regulation actually also play out in high eukaryotes? And for this, my former postdoc, Chelsea McLean, took advantage of a mouse model that we have in our institute of Jan Henneman, in the group of Jan Henneman Dannenberg, where HDAC1 is deleted in T cells in a conditional manner by the expression of the Cre recombinase under the control of the LCK promoter. So Cre is expressed in early T cell development, and then it deletes an essential exon in HDAC1. And so this is an overview of T cell development in the thymus, and LCK is expressed really early in thymic development of T cells. And then from early on to later, HDAC1 is deleted. And what Jan Herme had shown is that HDAC1 is a tumor suppressor in these cells. If you delete HDAC1 in the thymus, this leads to thymic lymphoma. Most mice will die within 20 weeks because of a thymic lymphoma. And so what Chelsea did, she took lymphomas that were isolated from this mouse model and compared this to lymphomas that were HDAC1 proficient, but P53 mutant. And so she took two cell lines, HDAC proficient and HDAC deleted, and confirmed that the HDAC knockouts have more acetylation of histone. And she found that indeed, like in yeast, there is an increase in K79 methylation, and it's especially evident from this dimethyl K79 blot. And we confirmed this by mass spectrometry. And what was actually quite exciting is that when she analyzed these cell lines that have increased methylation by HDAC knockout, these cells were more sensitive to the Dot1L inhibitor pinomidostat, or this epizyme compound. Like in black, we have HDAC1 proficient cell lines, they are insensitive. And in red, we have the HDAC1 deficient cell lines, and they were sensitive to Dot1L inhibitor, similar to the MLL rearranged leukemia cell lines. Right, so these results just show that this regulation is found in yeast, but also in the mouse, in a condition that also leads to thymic lymphoma. And that Dot1L inhibitors and that these cells that have increased K79 methylation are sensitized to Dot1L inhibitor. So they have a dependency on Dot1L activity for survival. And the Dot1L inhibitor, we found, induces apoptosis in these cells. So so this is an example of where we took data from basically determining the regulome of K79 in yeast using DNA barcoding, finding known factors, but also unknown factors. And one of these mechanisms is conserved from yeast to mouse with relevance for human lymphoma. And so having made the step to studying T cells, we thought, well, this is a unique opportunity to also look at what Dot1L does in normal T cells, because we know a lot about leukemia, and in this case lymphoma, but what really is the role of Dot1L in normal T cell differentiation? And this, what I will talk about next, is our work on Dot1L in normal T cells, and this is a collaborative effort between many people, especially the group of Heinz Jacobs, who's my T cell expert, my buddy in this case, and Elzo de Wit in our department helping us with some chromatin genomics studies with Teun. But the captains on the ship are these three people, Elisa Marie, Muhammad, and Farshid. What they did, they took the LCK Cre line and combined it with an allele of Dot1L where exon 2 is fluxed. And that means that if Cre is expressed in early T cell development in the thymus in this double negative state, the DN state, that there is no more functional Dot1L and no more K79 methylation. And we noticed that if we look at these mice, they do not develop lymphoma like the HDAC knockout, and we also didn't expect that. We confirmed the deletion by staining for K79, and you can see that in a knockout, the methylation is mostly gone. And we also developed a flow cytometry assay to look for K79 methylation in the nucleus of these different cell types. And this allowed us to determine at what state of development the K79 methylation is gone. And so here we sorted cells by flow cytometry based on surface markers to distinguish between subsets within the double negative stage where Cre recombinase becomes active, especially at the DN3 level. And we can see that the methylation is still, and K79 methylation is high in wild type cells in every state. And in a knockout of Dot1L, a conditional knockout, it is high in early development, and then it shifts to low in late development. But the loss of K79 is not immediate in the DN3 when Cre becomes active. It actually takes some time to start disappearing, and only in the immature single positive state it is completely gone. And this makes a lot of sense because in the absence of DNA of a demethylase, I just told you that cells need to replicate in order to lose K79. And so in essence, this means that we can really only study the function of Dot1 and K79 at a later stage. And so we focus mostly on the mature thymocytes in the thymus. And when we look at the different subsets and look at their abundance in the thymus, where in black we have wild type, in red we have knockout, and in gray the heterozygous knockout. It is clear that total cell numbers were not much affected, even later, except for maybe towards the end, where we have mature CD4 cells and CD8 cells. And when we had a closer look later in development in the thymus, this difference became very obvious, where we did see a very low number of CD4 helper T cells in the Dot1L knockout, and an increase in the number of CD8 cytotoxic T cells in a Dot1L knockout compared to wild type in black. And in fact, this became even more informative when we looked at K79 methylation by flow cytometry. In the CD8s at the bottom, we have high methylation in wild type and low methylation in knockout. And in the CD4 cells that were present in this model, basically they had not lost or had partially lost K79 methylation. And we looked at their genotype by PCR and found that the CD4 cells that did make it to the periphery, they had very low efficiency of deletion of Dot1L. They had escaped the deletion by the Cre recombinase. So there seems to be a very high pressure for helper T cells to maintain Dot1L activity for survival in this model. Okay, so Dot1L is crucial for CD4 T cells, at least in our mouse model. But we decided to look on the CD8 T cells because it would allow us to look at their function without Dot1L later in development. And so we analyzed CD8 T cells in the spleen and use cell surface markers to identify 3 different populations. It is the naive cells, the majority of T cells in a normal mouse. And when these cells are exposed to foreign antigen, they will differentiate to effector cells to execute their cytotoxic functions, and also to memory T cells for the long-lasting protection. And so when we look at this in a wild-type mouse, we can see they have mostly naive cells, some memory cells, and very little effector. In this, in our clean mouse facility, this makes sense. And then when we analyzed Dot1L knockout, the picture was completely different. Dot1L knockout mice, T-cell specific knockout mice, they have basically only memory phenotype cells. They have no more naive cells and a slight increase in effector cells. So there's an increase in these memory phenotype cells, but this is only looking at 2 cell surface markers, right? So what else have we done to study these T cells? So this is a summary of some key features. So the first of all is that these cells have high levels of T-beta-eniomas. These are 2 key transcription factors for memory and effector cells. So these cells really seem to be executing a program that is memory phenotype. However, we found no signs of any T-cell receptor activation by looking at the CD49D marker. So they didn't seem to have undergone any recognition of a foreign antigen. And this was confirmed by looking at the T-cell receptor repertoire. you know, if there would have been an infection, for example, we would have seen clonal expansion of certain T cell receptors. But in fact, in memory T cells in Dot1L knockout, there is a very broad repertoire, and we could not see any difference in TCR beta gene usage between wild type and knockout. So all these data suggested that Dot1L somehow prevents an differentiation towards memory cells in an antigen-independent manner. These cells seem to have differentiated without having seen any antigen. And so we call these cells antigen-independent memory phenotype cells, or AIM-T cells. And the reason why we call them AIM-T cells is because we're not quite sure what their precise origin is. In the field, people have seen these types of cells in different mouse models, in wild types, in humans. And so people have referred to these as virtual memory cells or innate memory cells. But their origin and their function is still very much under debate. And for example, these virtual memory cells can arise if T cells undergo more proliferation when there is, for example, loss of T cells in the periphery. Imbalanced TCR signaling is another mechanism that can contribute to these virtual memory cells. And also their function is still very much under debate. They could have bystander functions. They seem to infiltrate tumors more efficiently than regular memory cells. There's a recent paper about this in Nature Immunology that shows this. So these cells are very mysterious, yet potentially very important, but not much is known. And so, so we looked into this further to see if we could understand more about what Dot1L contributes to these, to forming virtual memory-like cells, and how Dot1L could, you know, do this by its epigenetic mechanism. But in essence, what we found summarizing this part is that in a normal mouse, there is a lot of naive cells that will egress from the thymus in the periphery. And then only when antigen is present, they will differentiate to memory cells. In the absence of Dot1L, this block is gone. And even without antigen exposure, these cells differentiate into these antigen-independent memory phenotype cells. So what are the molecular features of these cells without Dot1L that look like memory cells? And the first thing we did is make a gene expression signature of wild type, memory, and naive cells. And this allowed us to make a gene set of genes that classify memory cells in green, and gene set that classifies naive cells. And when we now plot wild type naive cells versus these Dot1L knockout memory cells, we can see that these gene sets, for example, in green, the memory genes, they are still expressed in a Dot1L knockout. And the naive gene set is actually more repressed in a Dot1L knockout. So this suggests that overall, Dot1L knockout memory phenotype cells, they do express the right genes, maybe not to exactly the same level, but overall, they seem to be executing their memory program. And so, but when we look at the gene expression in a bit more detail, maybe we can learn about, you know, what is going on in these cells. And so here we plot wild type versus knockout, and let's focus on this memory population. So RNA sequencing in wild type versus knockout cells, we have about 900 genes up and 800 down. And so what have we seen in this, these differentially expressed genes? And so, like I said, we, it is known that T cell receptor signaling is an important contributor to making virtual memory cells. And so we looked at T cell receptor signaling genes as a first test. And let's focus on the memory cells on the left, where we have wild type on the left and knockout on the right. And red means upregulated and blue downregulated. And we find that many of the TCR signaling genes, and both kinases, but also TCR components, are differentially expressed in a Dot1L knockout. And so this suggested that this could be maybe a mechanism that contributes to these antigen-independent memory cells. And we confirmed this at the protein level by flow cytometry. For example, the TCR levels are lower in knockout in red versus wild type in black. CD3 levels are lower, and also the CD8 levels are lower. So this really suggests that in the absence of Dot1L, there is a problem in expressing T cell receptor genes, in expressing co-stimulatory genes, but also in expressing some of the downstream signaling proteins in the cell. And so how does this relate to K79 methylation? So we did K79 dimethyl chip sequencing and first confirmed that if you look at where it occurs in the genome, it mainly occurs at the 5' end of genes in a mouse cell. And it was shown by the Yamamoto lab already some time ago that the methylation actually is in the domain until the first internal exon, and then it switches to K36 methylation, and this is the same in mouse T cells. And we found that many of the genes that are affected by Dot1L knockout, they actually contain K79 methylation at the 5' end of their gene locus, in the thymus, in memory cells, in naive cells. So potentially these could be directly affected by Dot1L, but obviously that we haven't demonstrated in this study. But it is true that Dot1L has a big impact on TCR signaling genes, and we believe that this could be one of the mechanisms that contributes to the formation of these antigen-independent memory cells. And it will be interesting to see whether this role of Dot1L is actually also part of the normal physiology that leads to the virtual memory cell formation. Okay, so then we asked, what is the relationship between K79 methylation and gene expression? Genes are methylated, and then when we lose methylation, they become lowerly expressed maybe. What is the general logic of this in the T cell? So we go back to this RNA sequencing data, and now overlay the RNA sequencing data with K79 dimethyl chip that we performed on the same cell populations. And again, let's focus on the top part where we look at the memory cells. And the first thing that we notice is there is a group, the black genes, they are downregulated in a Dot1L knockout. It's about 800 genes. And they are generally more highly expressed in this MA plot. They are localized onto towards the right end. So this is expression on the x-axis versus fold change on the y-axis. And now when we overlay this with K79 methylation score, and the red is high methyl, blue is low methyl, K79 dimethyl around the transcription start site of genes, we see that the genes that are downregulated, they tend to have high K79 methylation. So these could potentially be direct targets of Dot1L. But there's this other group of genes that is more towards the lower level of expression, towards the left, that is de-repressed in a Dot1 knockout. And they actually have no methylation or very low methylation scores. So we conclude that these genes must be indirectly affected by Dot1L knockout. And so then we ask, so can we maybe explain this? And is there perhaps in the genes that are possibly directly regulated and downregulated, are there any transcriptional regulators, repressors, that if they are reduced in expression, could lead to de-repression of these targets in Dot1L knockout? And so we searched for transcriptional regulators based on their methylation level, change in gene expression. We found 14 factors, and then one factor caught our attention, and that's EZH2, because it was known that knockouts of EZH2 also lead to a memory differentiation phenotype. And so we looked into EZH2 a bit further. EZH2 methylates H3K27 trimethyl. It's part of the PRC2 complex, which is the polycomp repressive complex 2. And it's known to repress developmentally regulated genes together with PRC1. And like I said, it has a known role in T cell differentiation. And we've confirmed that EZH2 is indeed methylated. We inspected the gene, and it has the normal methylation pattern. So this could be a potentially direct target, and loss of this could lead maybe to the de-repression of these genes we see in Dot1L knockout. And indeed, when we inspected this further, and here we have in, this is again, memory cells, wild type versus knockout, and on the top we have these de-repressed genes. And now in orange, I hope you can see this, we overlay the genes that are known to be de-repressed in EZH2 knockout in T cells. And you can see that there is overlap between the EZH2 de-repressed genes and Dot1L de-repressed genes. And this is also shown here, where we take the genes that are upregulated in EZH2 knockout in orange. They tend to be also upregulated in a Dot1L knockout. They're higher than the other genes. And so let's then compare this to K79, K27 trimethyl chip. And let's focus here on the, again, on these memory cells. It's a bit of a busy plot, but let's just focus on the top here. So here we focus on the genes that were de-repressed in Dot1L knockout. And compare this to a matched set of genes that has the same level of expression, but is not affected by Dot1L knockout. And what we see, they have generally a low level of K4 trimethyl. And these genes have no K79 methyl. Whereas expression matched genes, these are lowly expressed, they have K79 methyl, but at a low level. These genes actually have K27 trimethyl, whereas the control genes don't. And this is the same for the other subsets in naive and cells and thymocytes. So these genes that are upregulated in Dot1L knockout, they don't have K79 methylation, but they are enriched for K27 methylation. So we think that Dot1L, these are indirectly de-repressed BRC2 targets. And maybe just to highlight, in a study in human T cells, when we inspected the data from this study from Kagoya et al., they found that Dot1L inhibitor on human T cells actually also leads to reduced EZH2 expression. So this link between Dot1L and EZH2 in T cells could be conserved from yeast to humans. And so this is our current working model, where we have Dot1L regulates a subset of the genes that it methylates, about 900 genes, including EZH2 of the PHC2 complex. And this repression mechanism is important for normal T cell function. And then in a Dot1L knockout, this mechanism is lost, and EZH2 targets become de-repressed, and this compromises the effector function of T cells. And why do I say effector function? Because when we look at the function of Dot1L T cells, we find that they seem to be incapable of executing a normal effector response. Here, for example, we have a Listeria infection, and look at the clearance. After 3 days, we see lots of colony-forming units in the spleen. After 7 days in the wild type, the Listeria infection is cleared, but in the knockout, there are still many bacteria in the mouse. Also, when we do peptide vaccination and look for antigen-specific T cells, in a wild type, they accumulate over time after vaccination. In a Dot1L knockout, we see no expansion of antigen-specific T cells. And so, so this is why Dot1L forms, we conclude that Dot1L leads to the formation of antigen-independent memory cells. But these memory cells that have prematurely differentiated, they are not capable of executing a proper effector response. And I think our next challenge will be to understand how Dot1L really regulates genes, including EZH2, but also what the proteins are and genes are that are affected that do lead to this compromised effector function. So in summary, I showed you that Dot1L is important for CD4 T cells, and in CD8 cells, it forms like an epigenetic barrier towards differentiation. And if this barrier is absent, these prematurely differentiated cells cannot properly execute a CD8 effector response. And this is unpublished work that you can read more about in a paper in bioarchives, but also I would like to highlight a paper from the lab of Kolbe-Zev, where they look at CD4 T cells in a different knockout model and found kind of similar findings about differentiation. We clearly have some unresolved questions. What is the consequence of this late loss of Dot1L or Dot1L inhibitor? If you treat cells for leukemia, for example, will this also affect T cell differentiation? What is the role of Dot1L in other lymphocytes? And there are 2 papers on bioarchives that address a role of Dot1L in differentiation in B cells, basically showing very similar findings as we found in T cells. And then direct versus indirect effects, this is still a challenge for our enzyme because the methylation loss is very slow. It is intrinsically challenging to address what is direct and what is indirect. And then really, we don't still understand how the methylation affects gene expression molecularly. And I think this is still one of the big questions in the field. And for this, I think we will have to then find new ways to interrogate chromatin structure. And I would like to spend maybe 3 minutes or so to explain to you a system that we developed in budding yeast to look at chromatin structure in a different way. And then maybe this is a system that we can apply to understanding more about histomodifications and how they affect chromatin structure. And this method we call Epi-decoder. It is about decoding chromatin proteomes of a single genomic locus, and was led by these three fantastic people, Dipani, Tessie, and Ila, and with help of Kitty and Tibor. And so briefly then, what we would like to know, if there is methylation at the locus, in this red dot, for example, what proteins do bind there versus a locus that is not methylated? Can we find proteins that are bound there? And what current strategies are at the moment is to introduce handles at this locus by CRISPR-Cas or other methods, pull it down, do mass spectrometry, and try to find proteins in this way. But there is a challenge because purifying, say, a single locus of the genome is an enormous challenge simply by the factor of enrichment and also by the amount of material you need for mass spec. And so we thought maybe we can use our barcoding technology to contribute to solving this problem. And so I told you about Epi-ID, where we have barcoded yeasts and then combine it with knockouts. And now what I would like to propose is to use DNA barcodes, but combine it with tagged proteins so we can interrogate the binding of every protein in the yeast cell to the barcoded locus. And this then goes as follows. We have, again, this barcoded library. And now we combine it with a library of yeasts in which every colony contains a yeast where one protein is tagged with a tab tag. We use yeast genetics to combine this. And now we have a new library where basically the barcode identifies the barcoded, the tab tag protein. And again, we can pool the cells like with Epi-ID. We do cross-link the chromatin, and we do chip, not for K79, but for the tab tag. And now if a protein is bound in the vicinity of its barcode, the barcode will be IP'd. If the protein is in the cytoplasm, the barcode will not be IP'd. And so then we isolate DNA, amplify the barcode, sequence and count them. And now we get a barcode count for every tab tag protein. High barcode count means the protein in that cell was bound to its barcoded locus. And so now we conclude that barcode abundance is a measure for cross-linking of the protein or binding of the protein next to the barcode. And again, as a proof of concept, we use this Pol2 locus, where we have a promoter and a terminator barcode. And the terminator barcode is also next to an origin of replication, which will become relevant in a second. And when we look at how many proteins we see in this way, we find about 400 proteins that we can identify at this locus. And this is a huge amount. Surprised us a lot, but most of these proteins are proteins with a annotation in DNA or transcription in chromatin in red. But there's also a small subset involved in RNA metabolism or termination, cleavage of RNA, a bunch of metabolic enzymes, and some proteins that are like category other in this case. Lots of proteins, knowns and unknowns, and this is just an example of the type of data we get. So this is barcode counts, log2 scale for the promoter on the x-axis, terminator at the y-axis. And we see proteins that make a lot of sense, like histones are highly bound to both areas. We see H2AZ, which is known to mark promoter areas. It is more abundant at the promoter than the terminator. We see Pol2 components, elongation factors. We see proteins involved in initiation at the promoter end. We see factors involved in termination at the terminating end in green. And we see organ MCM proteins bound to the down tag, the origin at the terminator end. But then we also see other factors like heat shock proteins, SSA1 and SSA2. And we see factors that are initiation factors that are also cross-linking to that terminator. And I think what's really powerful about this method is that we interrogate all proteins together in competition using the same tag. And so we can actually directly compare their abundance. So we not only detect them, but I think their quantity is also informative. And so I think these heat shock proteins, SSA1 and 2, they cross-link very efficiently and maybe core components of the chromatin, if you will. And then as the last slide, I would like to show maybe more interesting data, because finding a map is interesting, but really it's all about the delta, right? How do proteins change in certain conditions? And then really you learn about what is relevant and what is not. And so one condition that we tested is quiescence. So Toshi gave a great talk about this in this series. And Christine in his lab took our libraries, grew these cells into quiescence, purified the quiescent cells and non-quiescent cells that are also there. And then we performed our Epi-decoder analysis on this population and compared it to log phase cells. And this, if you glance at the data, and this is about a subset of like 700 proteins that are all chromatin related or known to bind to these barcodes from previous studies, you can immediately see that log phase is the left, and then the next two lanes are quiescence. There are major changes happening in these cells. And let's just walk through a few examples to show what this tells us. So Pol2 components and elongation factors from log phase to quiescence are basically gone. But there's a few proteins like NHP6A and B, high molecular weight high mobility group proteins. They are increasing a lot. And so there's a few other factors that have been annotated as transcription elongation factors that also increase. So there's like a replacement of transcription proteins for other proteins. RPD3L, Toshi identified this as a repressor of global chromatin in quiescent cells. Indeed, this is increasing in quiescent cells. And we also see an increase in histone H1 and the high mobility group protein HMO1. Another example I think is quite interesting are these heat shock proteins like SSA1 and 2. They seem to switch their abundance in quiescent cells, but in general, there is an increase in protein folding machinery at the chromatin. And of course, at this moment, we don't know what this means, but I think it is very interesting. And then the last example is replication proteins. The down tag, the down barcode is next to a terminator, but also next to a origin of replication. In log phase, we found there is the MCM helicase and the ORC complex that recruits eventually the MCM helicase. And in quiescent cells, we see that the ORC proteins increase. And this actually goes together with a complete loss of the MCM helicase. And I think this means that for quiescent cells to re-enter the cell cycle and start replicating, they will have to re-recruit the helicase. And maybe this is a means for long-term survival of these quiescent cells. But in any case, I think here also it is important to say that all these proteins are IP'd in single tube, same tag, and their color basically immediately tells you about their relative abundance. And I think, for example, these heat shock proteins are light green. Their abundance is the same. Their cross-linking is the same as RPD3 or some of these elongation factors. So they are quite abundant compared to other proteins at the chromatin. Okay, so Epi-decoder is a way, in yeast at least, to directly and quantitatively report on chromatin binding of many proteins in parallel. It's an unbiased way and comprehensive way to look at chromatin proteome. And I think for the future, it would be very interesting to combine this with different conditions. For example, change the methylation level of K79 and ask what is now different. And also to decode different loci. We have already other loci that we can make barcoded libraries for by CRISPR-Cas. This is relatively easy. So this is, we can now look at many more places and really compare how Pol2, active, inactive, heterochromatin, Pol3, you name it, it's now possible. And I showed you that we can identify the dynamics of known components, but we can also identify unknown components, like maybe some of these metabolic factors. And for example, in a decoding of a Pol3 locus, we found recently an unknown protein that has not been annotated in yeast. That is a novel tDNA binding protein that we hadn't found before. And I think, if we do this from many loci, many conditions, we really will develop a better understanding of the interactions of the chromatin proteins in the cell, and then hopefully contribute to this bigger mission, right? That we understand the molecular encyclopedia of chromatin. Okay, thank you for your attention. I mentioned most of these people as we went along, and I also want to thank the people that make the projects work financially. And I would like to thank you for your attention, and I'd be happy to answer questions.
Thank you very much, Fred. That was a fantastic talk. It was a really cool display of what available technologies can be done and repurposed to do these really clever technologies. So I have one quick question, and I just want to remind people that if you have a question, we really prefer if you raise your hand and can ask it live. You'll jump the queue of all the other questions that we have written. So please go ahead and do that, and I can just give you the voice for that. I'll just ask you one quick question. Maybe I missed it, but during your Dot1 knockout in the T-aim cells, do you see a phenocopy if you inhibit PRC2? Do you also see this increase in the differentiation?
Yeah, I think this is a very important question, right? We have 800 genes that are affected, and one of them is PRC2. And so I think PRC2 is probably part of the response of these AIM T cells, but I don't think it can explain everything. And so we haven't done the experiment, but I predict that if we, for example, in PRC2 knockouts, they also form virtual memory cells, or these AIM T cells. But there the mechanism seems to be a little different. It seems to be a mechanism that involves cytokine production, whereas the Dot1L knockout, we've found out it actually works by a cell intrinsic mechanism. So I think we have to do this experiment, and maybe we should do this in a more simple system, in a more like in vitro system, to really look at this. But I suspect that it will not be that simple. But it is an experiment that we have to do. Yeah, I agree.
Okay, thank you. So I'm going to give the word to Purva. So Purva, whenever you're ready, go ahead and ask your question.
Hi, a very informative talk. Thanks a lot. I just have one question towards the last part of your talk, where you showed the interacting proteins to the chromatin.
Yeah.
The most interesting thing I saw was the polymerase itself was changing. That was on the top, right?
Yeah.
So a lot of other proteins that you saw there, they were all polymerase interacting proteins. In the crystal structure from the Kramer lab, you can see them binding there.
Yeah.
So shouldn't you be normalizing the whole heat map to the amount of polymerase change to know the real change of these proteins? Or do you think that it can never happen that a polymerase could still transcribe without these elongation factors?
Yeah. Yeah, I think this is a challenge that we haven't really been able to address. In how do we normalize the data? So what I can say is that in these experiments, there are still many proteins that are bound. Histones, we see metabolic enzymes, many of them cross-linked to the chromatin. And so there is a shift, and I think there could be like an off shift, if you will, of the average. But overall, there are still so many proteins bound that I don't think we have this explains all the data. I think maybe histones could be a way to normalize the data, but for example, in aging yeast cells, it has been shown that you also change the overall abundance of histones. It's not a fixed constant in the cell. So I think for now, you're right, we can, it is a relative change, and it is true that we see a loss of POL2, but also POL2 associated factors. Indeed. Yeah.
Thanks a lot.
Okay, thank you. So I'm gonna read one of the questions that we have from the public right now. So Deepak Jha, he says, great talk. I may have missed this, but do you know why there are so few CD4 plus cells? Is this a differentiation block owing to downregulation of pro-differentiation factors? Or is it that these mice make CD4 plus cells, but they end up dying due to the loss of Dot1?
Yeah. Yeah, so the CD4 cells is a mystery to us. And we haven't looked at this at all because, like I showed you, the CD4 cells that we do obtain in the periphery, they seem to have escaped the Dot1L deletion. And so this will always be a mix of knockout and wild type cells. And I think we were fortunate enough to monitor our T cells by flow cytometry to know for sure that the cells that we look at are really Dot1L knockout. And most of these CD4 cells are still wild type. And so we would have to find a way to split them up. Something else I think I didn't make this very clear. There is a study from Colby's CEST lab where they deleted Dot1L a little later in T cell development using, I think it was a CD4 cree. And they actually find that in this model, at a later deletion, the CD4 cells still form efficiently. But they also have a differentiation problem like we see in the CD8 cells. So I think this is a problem that occurs in the early T cell development, and that that problem is not present later in development. So Dot1L is not essential per se for CD4 T cells. And that model, I think, would be more useful for studying CD4 T cells. And that's there's a paper on bioRxiv that it's really interesting, the switching between the two subsets of T helper cells.
All right, he says, thank you for answering his question.
Thank you.
All right, so I'm gonna give the voice to Pallavi Mustafi. So whenever you're ready, please go ahead with your question.
Hello. Yeah. Yeah. Great talk. My question was, can you hear me?
I can hear you. Yeah.
Yeah. So this Dot1L starts showing its effect in the later stages of the T cell differentiation. So what do you think is regulating at its upstream? Means what is its activity? Why is it showing its activity in the later stages, and what is regulating it in the earlier stages that is inhibiting it? Do you think it is a?
Yeah. So I think maybe if I can rephrase your question is. So you find a phenotype of a knockout. Is this part of normal physiology? Is Dot1L, for example, regulated in T cell development to steer T cell differentiation? I hope I rephrase your question correctly. And I think in T cells, we don't really know. We looked at RNA levels, K79 methylation levels in different T cell stages, and it's quite constant. So at that level, there is no evidence that this is regulated to regulate T cell differentiation. In B cells, where we find a similar phenotype of premature differentiation and a link to PRC2, there we find that the cells that are dependent on Dot1L, they have higher levels of Dot1L, higher RNA levels of Dot1L and EZH2. So I think there is maybe evidence that it could be regulated in normal B cell differentiation, but in T cells we don't know whether which upstream mechanisms control Dot1L in steering T cell differentiation. It may not be regulated. Maybe it is simply part of the the core chromatin structure that allows other factors to execute their regulatory function. That is equally well possible.
Okay, thank you, Pallavi. Seems people say their questions have been answered. I mean, maybe I can just ask you one quick question too, if that's okay. So you were measuring the expression with steady levels of RNA-seq. Have you looked at if there's also the same correlation, or I mean, actually the lack of correlation of expression in the normal cells with H3K79 to see if there's any difference for nascent transcription?
Yeah, so we haven't done nascent, yeah, because we were at this point, I think we are not in the right position to do that. We took T cells in vitro, added Dot1L inhibitor, and then it takes a long time before you see a loss of methylation. And so I think the nascent is really to, it will be most helpful for us to see the direct effects. If you inhibit Dot1L, can you see fast effects? Yeah, I agree. It's still useful to exclude RNA stability processing from transcriptional defects. I think for that, we probably will still do this in our future studies. But I think we would like to solve this problem that, to understand maybe some of the fundamental questions. Can we somehow accelerate, K79 methyl loss in a way so that we can really get a grip on these direct effects on chromatin? Yeah. And then we would have to do nascent. Yeah. No, I think we will have to do nascent in the future. to be sure we talk about transcription, not about other RNA effects.
Yeah. Okay, excellent. Well, thank you very much, Fred. It was fantastic to have you here. Thank you so much for agreeing. And we're going to have some people, some trainees meeting with you soon thereafter. And thank you, everybody that attended the talk. I hope you have a good rest of the evening.
Thank you, everybody.